Welcome to DNA replication. What is DNA replication? Copying DNA. Or the chromosomes, right? Same thing. Chromosomes. Where does this happen? Where? Where is DNA? In the nucleus. For eucs, right? Procs, everything's in the cytoplasm. Doesn't matter. When does this occur? Interphase. Yes, remember that? Yes. What's specifically an interphase? S phase, the synthesis phase. Synthesizing DNA means making DNA. We want to copy each of the chromosomes to make a sister. Here's a chromosome. We pretend like it looks like a stick. We really know it's spaghetti, but we pretend like it looks like a stick. And what happens in S phase? We copy it. Now, what do we have? One chromosome, two sister chromatids. Remember all that? Good. I thought you did. Here it is, cellular DNA replication, 3 main steps. First, we have to unwind the DNA helix, right? It's a double strand. So if we were showing it, these 2 strands are hydrogen bonded together. We talked about that in the last capture. How do we do that? We break the hydrogen bonds. An enzyme called DNA helicase. You guys don't really need to know, but it breaks the helix. Anything with an ASE is always an enzyme. We also need DNA polymerase. ACE, an enzyme. Polymer, lots of DNA. Lots of DNA that's an enzyme, makes lots of DNA. Copies the DNA. Uses each individual strand, each one of these, break it apart, and then copy each strand. So each strand serves as a template, or what you use to do complementary base pairing. We call it semi-conservative replication because there's an old strand. We copy a new strand. We copy a new strand. Right. The hydrogen bonds are broken. Oh, I can't break them. My eraser's not working. There we go. We'll scribble them out. No hydrogen bonds. Now, these 2, one old strand and one new strand, are hydrogen bonded together. This is one sister, and this is the other sister, right? Each one of these is the stick. Sister, sister. Each stick is the double helix. So don't get that confused, right? This is the double helix, which means this is a double helix. This is a double helix. This is a double helix. That's all that means. Here we go. Here's a nice picture. This would be the template strand. Breaks apart with the helicase. Breaks these hydrogen bonds holding the 2 strands together. Create a new strand. Create a new strand. One is the template. One is the new. This would be a sister, and this would be a sister. And of course, we look to see a close-up of what's actually happening. Complementary base pairing. C's always go with G's. A's always go with T's. So these original template strands break apart. Hydrogen bonds break. They're gone. We add new nucleotides. Ooh, if it's a T, we add an A. If it's a G, we add a C. If it's an A, we add a T. If it's a C, we add a G. Woohoo! Template, new strand. Again, template, new strand. A, C, T, G, T, G, A, C. Right? 2 new strands. Sister, sister. Woo! So which of the DNA strands here are the template strands? And why are they called a template? A template is something you copy from. That's why they're called a template. Copy off of. Is that a word? Is that a phrase? Is that grammatically correct? I don't care. So which of the strands are template? This was a template, this was a template, which means this is a template, this is a template. Here's the template, here's the template. We copy, we do complementary base pairing. Idiot-proof copying. Whenever there's a T, you put an A. Whenever there's a G, you put a C. Whenever there's an A, you put a T. Wherever there's a C, you put a G. And who's the U? DNA polymerase. 2 new strands, sister chromatids, ready to move on. This is all S phase of interphase. We would go through the rest of interphase G2 and we would start mitosis or meiosis, depending on what type of cell it is. Where does replication occur? Yes, I like what you've done here. What did I say about these guys? Yes. How about some fun facts? Lots more enzymes are needed to do this than I'm telling you about. But the enzymes I listed are the only ones you need to know. 8 hours to replicate the billions of nucleotides in a human. On all of our, how many chromosomes? 46 chromosomes we need to copy. Right? From a stick to an X. Here's our stick, really spaghetti, to the X. All 46 of them have to do that. It takes 8 hours, 100,000 nucleotides per second. Whew! DNA polymerase is fast. And guess what? We could do this in a test tube. This is test tube replication. We call it PCR, polymerase chain reaction. We essentially do DNA replication in a test tube with just a little part of the DNA. Anytime we want to amplify, what is amplify? Make more, right? Crime scene investigations. Oops, that's okay. What the heck? Sequencing, paternity suits. You want to know what alleles you have for the tongue rolling gene? Are you homozygous? Are you heterozygous? You need to know. You do PCR. You can use a teeny tiny trace amounts of DNA at a crime scene. Trace amounts of saliva on a cup. In just a few hours. In a test tube using all the enzymes that you really need physiologically in a cell. Happen in a test tube. So we get a tiny bit of DNA. Original sequence. We add the primers just like you need in real replication, which I didn't tell you about, so anyways. And then we add a polymerase, just like in the cells. And then we do this over and over and cycle through and through and through until we can amplify lots of DNA in a test tube. So most molecular genetics and genetic engineering is based on a physiologically relevant mechanism. So PCR is DNA replication in a test tube. And there you have it. There's part 2 of chapter 8. I hope you enjoyed it as much as I did.